but we're able to talk about some really cool things after we talk about mitosis, which is really a process of asexual reproduction, and meiosis, which is used in the process of sexual reproduction. So what this is allowing us to do is approach our discussion of genetics and how it is the human genome actually works. I feel like I'm going through cellular respiration a lot right now, trying to erase all of this stuff. Okay, so this begins chapter 8. Let me get that PowerPoint up on the projector really quickly. Yeah, there'll be a little bit of chapter 8 on the exam, but this is mostly going to be introductory kinds of stuff, so it won't be too in-depth. Okay, so chapter 8 deals with mitosis. And mitosis is largely taught incorrectly, unfortunately, because we talk about mitosis in the context of cell division. In fact, many people offer that definition for mitosis, that it's the division of a cell, and it's really not. Really, mitosis is just the division of the nucleus, one part of the cell. And that's a part of a larger cell cycle. Now, there are entire groups of organisms that only use the mitotic process to go through reproduction. The best example of those are the bacteria. They don't sexually reproduce at all. They reproduce asexually. Mostly, bacteria are unicellular organisms. And they will divide their cell, they'll go through mitosis and then cell division to create a second individual that is genetically identical to itself. Now, before that can happen, we need to have the replication of DNA and the dividing up of all the cellular machinery so that those two daughter cells, and we always refer to the cells produced by mitosis in a feminine sort of way. They are daughter cells. They come from a mother cell. Those two daughter cells are genetically identical to each other, and they're genetically identical to the mother cell. Now, we've already talked a little bit about plasmids. And prokaryotes, bacteria specifically, have an interesting type of chromosome. They have a circular chromosome. Ours are all linear, and theirs are circular. And those circular chromosomes are called plasmids. And like our DNA, it's double-stranded. So it looks something like this. It's just a big circle. And these big circles, these plasmids, are cool for a whole bunch of reasons. And hopefully we'll be able to talk a little bit about DNA technology and cloning and things like that, because plasmids are used all of the time. So there is an outer strand of DNA, and then there is a complementary inner strand of DNA. So just like ours, their DNA is double-stranded. When plasmids want to replicate themselves, or when a bacterial cell is getting ready to go through mitosis, what happens is an origin of replication appears within that plasmid. And really it just looks like a loop, kind of like this, where part of the complementary strand of DNA detaches from its complement. And then some cellular machinery comes in here and it starts creating another complementary strand on the top, perhaps in this direction, and a complementary strand on the bottom, in this direction. And then that process continues until both of those complementary strands have been completely replicated, and you get what was one plasmid, two plasmids, right? So one sister plasmid would look like this. I know I drew it smaller, but imagine it's about the same size. And the other sister plasmid would look like this. where you get the original template strand with a new complement inside. That is, this one is replaced by a new complement. And then what was the complement to the original outer strand gets a new complementary outer strand. Like that. So the point here is, and this was a really tedious way for me to present this idea, the DNA is replicated. We have 2 copies now of the exact same DNA. Mac, did you have a question?
Yeah, so the gap in the prokaryotic cell, is that where the origin of the replication is?
Yep, that gap is called the origin of replication.
Okay, thank you.
You bet. Okay, so once the DNA has been replicated, and do prokaryotes have a nucleus?
No.
No, they don't. So this is really, really simple for them. Once that happens, they're able to just kind of divide up all their ribosomes and proteins and split them into 2 cells. And that process of splitting the one cell into 2 is called binary fission. Sorry, I've advanced. Binary fission. Okay, so we had one cell. We now have 2 daughter cells that are genetically identical to each other. Again, it happens all the time in bacteria, and it can be a really fast process. So E. coli is a bacteria that we culture quite a lot, and we use it a lot for cloning and DNA technology. And the generation time for a E. coli cell is like 30 minutes. You can get a new E. coli cell about every 30 minutes, which is crazy. So it's estimated, right, that if they were given an infinite amount of resources and you start with a small bacterial colony of E. coli, like a cell, and you let it divide as much as it wants, within 2 days, the mass of that one colony would outweigh the mass of the Earth. It just replicates incredibly fast. But prokaryotes aren't the only organisms that only go through asexual reproduction. Fungi do it as well, and those are eukaryotes like us. Trees do it all the time, or plants do it all the time. Eukaryotes just like us. In fact, most stands of quaking aspens, they're all clones of each other, and they've reproduced via mitosis and cell division. There are some animals that are only known by females. So there's some species of lizards, for example, where there are only females and they're able to reproduce via mitosis. Some insects do exactly the same thing. Okay. So let's talk about our mitotic division. First of all, our chromosomes are a little bit different than those of prokaryotes, right? We have linear chromosomes. Really what that means is we've taken our huge long, 2-meter long DNA molecule and we've broken it up into discrete segments. And how many chromosomes does a human have? 46, right? We have 46 chromosomes. And what that means is we took that huge molecule and broke it up into 46 discrete units. But that huge molecule is really unwieldy. It's difficult to move around. And so what we've done is we've associated it with some proteins called histones. Through the process of mitosis, what we want to do is to take those 46 chromosomes, replicate them all, and split them into 2 daughter cells that are genetically identical to the mother cell. This is how all non-reproductive cellular division occurs. That is, this is the kind of cell reproduction that occurs in somatic cells. Somatic cells are all of your body cells except for where gametes are produced. So in females in the ovaries, in males in the testes. Outside of those regions, all cell division occurs via mitosis and cytokinesis. There is another cell division process that I know you've heard of as well, and that's called meiosis. And this is a cell division that occurs within those gamete-producing tissues. And its purpose is quite different than mitosis. Ultimately, what we want to do through meiosis is produce 4 daughter cells that have half the number of chromosomes that are all genetically unique. So that only occurs in our reproductive tissues. Now, that's the last we'll say about meiosis, I hope, until the next chapter.
What'd you say after in meiosis we want 4 daughter cells? We want half of what?
That each have half the number of chromosomes. So for us, each gamete that we produce would have 23 chromosomes.
Oh.
Right? And each of those chromosomes is going to be genetically unique. Okay. This will be the last slide that I cover for the exam. Mitosis is a part of a larger cell cycle. And the cell cycle itself consists of 3 discrete phases. But each of these 3 discrete phases has its own subphases. And so within the cell cycle, there is interphase. And interphase is not a stage of mitosis. Interphase is where we do all of the preparatory things for cell division, and it consists of itself 3 discrete phases. There's a phase called G1, a phase called S, and a phase called G2. Now, in the G1 phase, and we call them G phases, that G stands for gap, and we call them gap phases because when we originally were looking at cells in these interphase stages, it didn't look like anything was going on. They were just like in some sort of stasis. So it was a gap in between cell division and the S phase. But what we know now is during the G1 phase, we begin replicating all of our organelles. We know that each daughter cell has to have a Golgi apparatus. And we know that they need to have ribosomes. And we know that they need to have endoplasmic reticulum and things like that. So in gap 1 phase, we start replicating all of those organelles. In the synthesis phase, we actually replicate our DNA. And that's going to be the topic of discussion after we talk about genetics. It's like chapter 11 and 12. We talked about how DNA is synthesized. And then in G2 phase, after all of the DNA has been synthesized, all of our organelles are going to be completed. All of our proteins get replicated, like those proteins that are floating around the cytosol that are used for glycolysis. All of those will be replicated. And then our we don't use cell phones, right?
I don't have my phone.
I wasn't, that's not you, Sydney.
I'm like, oh.
So in G2 phase, all those organelles are replicated, and then we replicate our mitochondria. Now mitochondria replication is a little bit different, right? Because mitochondria has its own DNA, and all of the proteins involved in like cellular respiration are going to be constructed by the DNA inside the mitochondria. So after interphase, all of the preparation has been made that's necessary for nuclear division. And then we enter the end phase. And so that's phase 2 of the cell cycle, our end phase. And this is actual mitosis, right? So this is where we go through prophase and prometaphase and metaphase, anaphase, telophase. And the nucleus ultimately ends up dividing. And then after the end phase, we then do actual cell division, and that's the C phase, or cytokinesis.
So that's the 3rd phase? Or is that with, that's the 3rd phase? Okay.
Right, so the 3 phases in the cell cycle are interphase, M phase, and the C phase. Interphase has 3 phases within it, which is the G1 phase, the S phase, and the G2 phase. Okay. But yeah,
As far as what is going to be on the test, what should we do? This chapter, all of it?
yeah, stuff we've gone over so far. Certainly know what's going on in each of the phases within interphase. know what the purpose of G1 phase in is. That's beginning to replicate our organelles, right? Know what the purpose of the S phase is, replicating our DNA. Yeah.
Can
M
you,
phase,
sorry.
go ahead.
Can you repeat what the M phase and the C phase, like
Sure.
what they were? Yeah.
So the M phase, this is mitosis. This is where nuclear division occurs. That last most important organelle within our cells gets divided up here. And then cytokinesis, the C phase, that's where the cell actually divides. All of the organelles and nuclei have migrated to opposite ends of that mother cell. And then for animal cells, it just kind of pinches off in the middle.
Thank you.
You're welcome. Other questions? Okay, so you guys, if you have questions, I have office hours, of course. You can talk to me before and after lab tomorrow. We'll be doing our cellular respiration lab, I hope. If I don't see you in office hours or in lab, then I guess I will see you for the exam on Wednesday. Lab's not optional, by the way.